We're going to be starting with the second part of biological molecules. In this part, we're going to be talking about lipids and about proteins. I will start with an introduction about solubility in water. Materials that dissolve in water, we usually call them hydrophilic. A hydrophilic material is the one that could dissolve in water. Solubility depends on the nature of the solute. So for instance, table salt dissolves well in water. That's because of its ionic nature. Table salt, or NaCl, contains positive and negative ions, and these are quite hydrophilic. Usually all materials that are charged dissolve well in water. There's another option for a material to be hydrophilic. Materials can also be hydrophilic if they contain oxygen, nitrogen. That's because the presence of oxygen and nitrogen in small molecules turns those molecules into polar molecules. Polar molecules are very well soluble in water. Lipids like oil, on the other hand, are insoluble in water. They actually repel water. For that reason, we describe lipids as being hydrophobic. A hydrophobic material is usually non-polar, without charges, and it actually repels water. So for instance, materials that are made up of carbon and hydrogen only, usually we call those hydrocarbons, are usually water-hating or hydrophobic. That's how the word phobia comes from. It comes from the fact that these materials hate water. So that's where the suffix hydro, that's the water part, and phobia, which is hating of water. Lipids are mostly made up of carbon and hydrogen. They don't contain nitrogen or oxygen, and they don't contain charges. So that's why they are not hydrophilic. They are actually hydrophobic. We're now going to talk about the role of lipids in living organisms. Most living organisms contain lipids to store energy. For instance, mammals like humans store energy under the skin in the form of fatty cells. So in this picture we see fatty cells, so we call those adipose tissue, that stores energy for a long time. We took last lesson starch and glycogen. Fats and lipids usually store energy for a longer period of time than starch and glycogen. So that's why we call lipids as long energy storage. This type of long energy storage is called triglycerides. We're now going to have a quick chemistry introduction. To learn how lipids form, you really have to have basic knowledge of bonding and the types of hydrocarbons. Hydrocarbons actually fall into two categories. Hydrocarbons could be saturated or unsaturated. Saturated hydrocarbons are the ones that are entirely made up of carbon, and hydrogens, where every single carbon is attached to its neighbor with a single bond. So there was no carbon-carbon double bonds in saturated. Unsaturated hydrocarbons are the ones that actually have the carbon-carbon double bonds. So you could have a single double bond between the carbons, we still call it unsaturated, or you could have plenty of those carbon-carbon double bonds, we call it unsaturated. So unsaturated in general is any chain of hydrocarbons that contains a double bond. The second piece of chemistry that we need to understand here is the chemical groups. So there's one chemical group that we will need later on in today's lesson, which is called the alcohol group. An alcohol group is made up of just oxygen and hydrogen. We call this group actually a hydroxyl group. An acid group is always made up of carbon attached to an oxygen with a double bond with an OH. So this acid group is found in fatty acids and is also found in amino acids. So that's a general carboxyl group which you find in both fatty acids and amino acids, or in generally all organic acids. Finally, I want to talk about a bond that forms between the hydroxyl group of alcohol and the acid group of carboxylic acid. An ester bond forms when an acid group, which is COOH, binds to an alcohol group. The binding here is similar to the one we took with carbohydrate. This bond involves the removal of a water molecule. So you basically remove the OH from the acid group and the hydrogen from the alcohol group to form an ester bond. Ester bonds look like C double bond O with an oxygen. So you could link up an alcohol with an acid with this type of bond that we call ester bond. We are now going to use the chemistry knowledge to build triglycerides, the energy storage that we've previously seen. To build a triglyceride, you will first need an alcohol. This is a special alcohol. This alcohol is called glycerol, and it's special because it got 3 OH groups. Those 3 OH group will now be used to attach to acids, fatty acids. This will form triglycerides. So the tri part here is because we are going to attach 3 fatty acids. So the fatty acid, which is shown here, will attach to the glycerol. So the fatty acid, as you can see here, is made up of a long chain of hydrocarbons. So this is the hydrocarbon chain. Sometimes in the exam they show the hydrocarbon chain as a zigzag. So the zigzag represents a chain of hydrocarbon. You can actually use this zigzag form to represent a hydrocarbon chain. Other than the chain of hydrocarbons, fatty acids, as we've seen earlier, they contain this group right here. That's the acid group. The OH, or the hydroxyl group, or the alcohol group of glycerol, is shown here. Together those two groups will condense by the removal of a water molecule. So we remove this water molecule from here to form an ester bond. You can now see the formation of the ester bond. So the ester bond is this part, which belongs to both the acid and the alcohol. We still have room to attach 2 more fatty acids, and this will result in the formation of a triglyceride. Here is the triglyceride. So it's made up of the glycerol part. There is ester bonds in the middle and chains of fatty acids that are made up of hydrocarbon. Notice that the formation of one triglyceride molecule involves the removal of 3 water molecules by condensation reactions. One way to show triglyceride is in this format. So you show the glycerol like a head, and you show the fatty acid as 3 tails. So this head here represents the glycerol and the ester bonds, and the lines represent the fatty acid chains. Triglycerides could fall into 2 categories depending on the nature of their fatty acids. Fatty acids could be saturated, where every single carbon-carbon bond, as we can see here, they're all are single. So that's where saturated is. So saturated means it basically has the maximum possible number of hydrogens per carbon. Saturated fats are usually solid at room temperature. Very good example of this is butter, and most animal fats are actually saturated. Saturated fats turn to have like straight chains. The fatty acid chains are usually straight. So this makes those chains pack on top of each other very well. So the stacking of saturated fatty acid is very well with close attachment between those chains. This results in strong forces of attraction between those chains of fatty acids. You could see here in this picture, I'm trying to show you how saturated fatty acids, being straight chained, can interact well with each other. This kind of interaction between hydrophobic chains of fatty acids, we usually call it van der Waals forces of attraction. Those van der Waals forces of attractions are responsible for the high melting point of saturated fatty acids. Saturated fatty acids take a lot of heat to break those van der Waals forces of attraction. Unsaturated fatty acids have at least one double bond between the carbons. So the presence of a double bond changes the shape of the fatty acid chains. So instead of being straight chains, unsaturated fatty acids tend to have a bent shape. The presence of the double bond and the bent shaped fatty acids cause these fatty acids not to be able to be stacked on top of each other. So the stacking won't be as perfect the way we've seen with saturated. With saturated fatty acids, they used to be packed closely, straight chained fatty acids. Here, every time you have a double bond, you end up having a kink or a bend in the shape, and that results in distance between the fatty acids. So the van der Waals force of attraction here are going to be weaker, and that results in lower melting point of unsaturated fatty acids. So this is why unsaturated fatty acids like olive oil is liquid, while saturated fatty acids are usually solid like butter. We will now move to study amino acids. Amino acids are the building blocks of proteins. Every amino acid is made up of three major parts. There is the carboxyl group, which is found here, and the amine group, which is found here. In addition to those two groups that are standard for every single amino acids are different by their R group. So this R group is different from one amino acid to another. There are 20 different amino acids in nature. Each amino acid is different by a different R groups. We could actually classify those 20 amino acids into four major categories. Some amino acids are charged positive because of the presence of a positive group in its R groups. Some other amino acids are negative. Amino acids could be also hydrophobic if the R group is made up entirely of carbon and hydrogen. Finally, the last group is few amino acids that contain the element sulfur. So amino acids that are positive or negative are usually hydrophilic. Those that are made up of just carbon and hydrogen are hydrophobic, just like other organic molecules. Amino acids can also form bonds together by means of condensation reactions. So right here we got two amino acids, and you could see we've pointed the acid group on the left to the amine group in the other amino acids. These two groups will undergo a condensation reaction, so we're going to remove the OH group from the acid and the H group from the amine group. This will result in the formation of a water molecule. So the removal of the water molecule by a condensation reaction will result in the formation of a bond that we call a peptide bond. So peptide bonds are the ones found between amino acids. Peptide bonds look like carbon, oxygen, and nitrogen, hydrogen attaching to amino acids. Proteins are only functional when they take a 3D shape. So in this picture we see a necklace. Each one of those beads represents one amino acid. These amino acids are attached by peptide bonds. Now, a long chain of amino acids attached by peptide bond is what we usually call a polypeptide. Polypeptides are not proteins until they take a 3D shape. One way for a polypeptide to turn to protein is to start the process of folding. So those amino acids will now start folding on itself to form what we call secondary structure. The polypeptide chain itself is what we call a primary structure. The folding in this shape, we call this shape usually an alpha helix, is the secondary structure. Secondary structure will still fold on itself until it takes a proper 3D shape. This is what I usually call a tertiary structure. You could see here both forms of secondary structure. So the one here, that's what is known as the alpha helix, and the beta sheets. Both alpha helix and beta sheets are forms of secondary structure. Both forms of secondary structure result from hydrogen bonds. So hydrogen bonds are the ones responsible in the folding of the polypeptide chain into this particular two shapes. Here's another look at the secondary structure. You could see on the left the alpha helix. Another possible secondary structure is known as beta sheets. Beta sheets or alpha helix are foldings of the primary structure by means of hydrogen bonds. So both forms of secondary structure, whether if it's the alpha helix or the beta sheets, are results of hydrogen bonds that occur within the primary structure. The alpha helix or the beta sheets will fold further to form the tertiary structure. Tertiary structure includes those alpha and beta sheets, right, into a 3D shape. At this point, I could call this 3D shape a protein. However, in some cases, you end up having more than one polypeptide chain. This is known as a quaternary structure. So, tertiary structure is a protein. So, getting to this level alone is good enough to be a protein. There are several types of bonding that are responsible for the 3D shape of the protein in its tertiary structure. One type of bonding is hydrogen bonds. So hydrogen bonds do exist in the secondary structure and the tertiary structure. Here in this case we see an R group that has oxygen attaching to an R group that has hydrogen. This could also occur between R groups with nitrogen and hydrogen. Van der Waals force of attraction occur between hydrophobic R groups. So if there's an R group that is made up of carbon and hydrogen and the same type of R groups, those will interact by Van der Waals force of attraction. Third type of bonding, which is quite strong, are disulfide bridges. Disulfide bridges occur between amino acids that contain sulfur with each other. Those disulfide bridges are quite strong that they could actually bring very distant parts of the polypeptide chain together. One last type of bonding is the one that occur between positive and negative R groups. This type of bonding is known as ionic bonding. So ionic bonding is the fourth type of bonds that are responsible for the 3D shape of proteins. We could classify proteins into two major groups. One group is called fibrous. Fibrous proteins tend to be large and linear in shape. Fibrous proteins are usually structural proteins that support organisms. The other class of proteins is called globular. Globular proteins tend to have spherical shape and they're quite smaller than fibers. Globular proteins are usually the ones that carry out functions. So hemoglobin is a globular protein, so are all enzymes, hormones, and antibodies. In terms of R groups, fibrous proteins tend to have hydrophobic R groups. This makes fibrous proteins always insoluble in water. And this is related to their function because fibrous proteins like nails or hairs have to be insoluble in water to provide support. Globular proteins, on the other hand, have hydrophilic R groups. So those R groups here on the surface of hemoglobin have to be all hydrophilic so that the protein would dissolve in the cytoplasm of red blood cells. On the other hand, if you look closely inside, the R groups inside hemoglobin or all other globular proteins have to be hydrophobic. Being hydrophobic inside and hydrophilic on the surface helps to maintain the shape of globular proteins. Here are some examples of fibrous proteins. So one example of fibrous protein is keratin, which makes up hair and fur in the animal kingdom. Claws and nails are also made up of fibrous protein. Silk is another example. Proteins lose their 3D structure whenever they're exposed to environmental factors such as heat or pH. Heat causes particles to vibrate, and when particles in a protein vibrate at a high speed, this results in the breaking of hydrogen bonds. So the effect of heat on the 3D shape and causing a protein to denature is because heat breaks hydrogen bonds. On the other hand, pH results in the change in the overall charges of the protein. pH messes up the positive and negative charges on the R groups, and this also results in the breaking of specific bonds in the 3D structure of a protein. These bonds are the ionic bonds. So heat breaks hydrogen bonds while pH breaks the ionic bonds. The Biuret test is used to test for the presence of the peptide bond. Biuret reagent is added to the protein sample. If the color turns violet or lilac, then there should be proteins in the sample. We could take this test to another level and confirm the results by using a colorimeter. The colorimeter could be used to provide data about the intensity of color. So by taking the test tube that contains the protein sample and placing it in the colorimeter, the device here could provide us with data about the intensity of color by means of data about the transmission or the absorption of light as it passes through the sample.